Hi, this is Dr. Lauren Launen, continuing with a lecture set on DNA synthesis. So we just looked at replication forks. Now let's talk about how synthesis begins. So synthesis is going to begin, of course, at the origin of replication site, which we've already discussed. And those are sites on the DNA that recruit or attract the DNA replication enzymes. And so what does that mean? In order for synthesis to begin, what has to happen is you have to have the DNA replicating enzymes, shown in color here, land on the origin of replication. They need to open the double helix, and before that can happen, you need to relax the double helix as well. You need to undo what is called supercoiling. So topoisomerases are going to relax the twisting forces known as supercoiling, and then helicases are going to actually open the replication fork by breaking the base pairs that would anneal or keep together the double-stranded DNA molecule. Single-strand binding proteins, which are shown here in a darkish blue, are going to be responsible for keeping the replication fork open. And that is going to happen at the beginning of all DNA replication. So again, the single-strand binding proteins, shown here in a dark blue, are what keep the double-stranded molecule from re-annealing or re-base pairing, a spontaneous process. All right, so we're going to talk first about synthesis on the leading strand and then about synthesis on the lagging strand, but of course both are happening simultaneously during DNA synthesis. Leading strand synthesis is synthesis of DNA that is moving into the direction of the replication fork. In this figure, this molecule here, red and kind of goldish yellow, is the new strand that is the leading strand. It is being synthesized from left to right, which is from 5' to 3'. This is the 3' end of the new molecule. That means that the template strand, which is in light gray, is oriented so that this side is the 3' side, this side is the 5' strand, so that the two are anti-parallel to one another. So what's going to happen once the replication fork has been created and it is being maintained by these single-strand binding proteins, the first thing that will happen is an enzyme called primase will act to synthesize an RNA primer. So this little yellow region is complementary to the template, but it is made out of RNA nucleotides. All primers in the cell are made out of RNA. And that's the 5' end. This is the 3' end. If we were able to zoom in on the nucleotide at that position, the 3' carbon with its associated hydroxyl or OH group is pointing out in this direction. Okay? So synthesis is going to proceed this way. So once the primer is made by DNA primase, then what is going to happen is DNA polymerase 3, which is shown here in the green blobs, and also with this blue blob, okay, and the connectivity is not shown just to keep the figure clean, but it does exist, okay? So all of these proteins are in fact, not this, but these two clumps of proteins are in fact connected, okay, in real life. So here's DNA polymerase acting on the leading strand. It's catalyzing the formation of phosphodiester linkages. The nucleotides are going to be floating around in this entire environment and at relatively high concentration, and the appropriate nucleotide will land and base pair with the template. So it's base pairing between the template strand and the new nucleotide that controls the identity of each nucleotide being added. Okay? Then DNA polymerase simply has to catalyze the formation of the phosphodiester linkages that are holding all these little nucleotides that look like little teeth on a comb here together. And DNA polymerase, of course, always works in a 5' to 3' direction. So it's DNA polymerase 3 doing this. This is the major extension stage. It's doing most of the work. Once synthesis of the leading strand begins, it moves continuously in the direction of the replication fork. Okay, so the replication fork is going to continue to open as synthesis progresses. And you should note that on the leading strand, you only require one primer to begin synthesis. So I didn't say that before, but if you look at this cartoon, it illustrates it. Note where the replication fork is here, and then note that a moment later it's further to the right. So this replication fork is going to keep traveling this way, as synthesis progresses, and you'll get continuous synthesis of this leading strand in this direction till it gets to the end and it's finished. All right, so what about the lagging strand? So lagging strand synthesis is a little bit more complex because it is happening in the direction moving away from the replication fork, and yet it is being done by the same enzyme synthesizing the leading strand. So that enzyme isn't made out of like a rubber band. It can't just stretch and stretch and stretch. It has to stay in the replication fork. That means that it's going to work by starting and stopping, starting and stopping, starting and stopping in order to keep the DNA polymerase III in the replication strand. This means that synthesis of the lagging strand is happening in bursts, which is called discontinuous synthesis. This is a mechanism worked out by a scientist with last name Okazaki. Hence, we call those multiple short sections of DNA Okazaki fragments. And this is going to sort of challenge the DNA replication system a little bit more. It's going to have to work a little bit harder. You're going to have multiple primers. Therefore, the DNA polymerase or the DNA primase is going to have to work multiple times. This is a picture of this. Here is a photo of Reiji Okazaki and his wife Tsuneko. They were a husband and wife science team, and this is the group, this is the team that discovered how synthesis on the lagging strand works. This photo was taken in '75, and I think that was about the time that that discovery was actually made. And here is a photo of Arthur Kornberg, who of course discovered DNA polymerase I, and his wife Sylvie. They're also scientists, so this was kind of a neat example of two teams of husband and wife scientists that made some really important discoveries in the world of DNA and RNA replication. All right, so let's think about leading and lagging strand synthesis again. So we just talked about leading strand synthesis, which is continuous, and that would be the synthesis on the top strand here. Here's lagging strand synthesis on the bottom strand. It's defined as being synthesis moving away from the replication fork, right? Here's the replication fork. It's traveling from left to right. Here's the lagging strand synthesis. It's moving from right to left. And so over time, what will happen is, since the replication fork itself is moving, you'll get this gap created. And that's not going to work, right? You don't want to have a chromosome that has holes in it. So how is this gap going to be filled in? And the answer is that the DNA polymerase that's doing all of this work, it's synthesizing over here and it's synthesizing over here, it's essentially going to skip back and backfill. And then a moment later, the same scenario will happen again. It will skip back and backfill, skip back and backfill. So this is colored just a little bit different, but this is, excuse me, excuse me. This is how synthesis of the lagging strand is going to work, and we're seeing a few more of the proteins involved. We still have topoisomerase doing its work. We still have helicase doing its work. We have an RNA primer doing its work, or RNA primase doing its work. Here's the RNA primer. So a primase will have to synthesize an RNA primer to begin, just like on the leading strand. Right? So we've got the primer, and then DNA polymerase III is going to carry on from the primer. It's going to extend from the primer, always adding nucleotides to the 3' carbon at where it's got a hydroxyl group overhanging as it synthesizes from 5 to 3. And here's the synthesis happening on the leading strand. It's all related. These two are tethered or tied together. Okay, so this protein piece cannot get too far away from its other half, okay? So we've got a primer and then synthesis. We had a primer and synthesis. So this was synthesized, and then a moment later it skipped up and started again. Primase worked to make an RNA primer, and then DNA polymerase III continued from the primer all the way up to this first primer region. Each of these is referred to as an Okazaki fragment. Okay, so what will end up happening is on the lagging strand, you'll have these multiple segments of DNA, each with an associated RNA primer. Chromosomes are not made out of RNA, so these RNA primers need to be removed, and they need to be replaced with DNA, and that is accomplished by DNA polymerase 1, the polymerase enzyme that Arthur Kornberg discovered. And here it is chewing out the RNA nucleotides and then replacing them with, it's going to replace them with DNA nucleotides, and that will leave new nucleotides in the primer region, but they won't be closed together on the outsides with phosphodiester linkages. So the last thing that has to happen, kind of a polishing step, is the enzyme DNA ligase has to come in and it has to connect together the DNA at the primer region with the flanking DNA on either side through formation of a phosphodiester linkage. So if I were to ask you a question, what is the chemical reaction that DNA ligase can perform, you would say formation of a phosphodiester linkage. This will happen at the primer region. Similarly, if I said, what is the catalytic activity of DNA polymerase 1? You could also say formation of phosphodiester linkages at the primer region, but that would be in the middle. DNA polymerase 3, not shown here, well, actually shown here. All right, quick interruption from the phone. Sorry about that. So back to wrapping up our discussion about the synthesis of the lagging strand. So I think I just went through telling you guys the role of DNA polymerase 1, which is to replace the RNA at the primer region with DNA. You will, of course, have to form phosphodiester linkages between the new DNA nucleotides that are being added at the primer region. They're all in here at this point in time. Then you have the action of the protein called DNA ligase, and its job is to come in and close those last phosphodiester linkages that are in fact going to be on either side at the primer region, or sorry, not either side, on one side at the primer region. And that will ensure that this strand is intact. There are no little gaps or bumps in it. Okay? And so essentially on the lagging strand, what's going to happen is all of what I just described is going to happen over and over and over again. So as the replication fork opens from left to right, primase will begin, DNA polymerase 3 will extend, then it will skip back up and do that again. And every time it skips back up, it creates the time and the room for the enzymes DNA polymerase 1 and then DNA ligase to do the job of replacing and polishing at the primer region. And that will happen at the same time as the synthesis that is occurring on the leading strand that's kind of blocked out up here. And all of this will continue until this replication fork bumps up into either the end of the chromosome or synthesis coming at it from the other direction. And that concludes our discussion of the leading and lagging strand synthesis.